Hello everyone, I am Divya Dhanke from Shivajirao S. Jondhale College of Pharmacy, Asangaon, studying in second year B Pharmacy. Today I am going to explain the topic from Pharmacognosy and Phytochemistry first B.Pharm 4th semester, that is edible vaccines, under the guidance of Dr. Nitin Mohire as a professor and principal of our college. Here, first we see the introduction about edible vaccines. Edible vaccines are transgenic plant and animal based production of those that contain agents that trigger an animal in response. In the simple terms, edible vaccines are plant or animal made a pharmaceutical. Edible vaccines contain DNA fragments from the original pathogen. History of edible vaccines. The concept of edible vaccine was developed by Zheng in 1990s, head of Department of Plant Biology at Arizona State of University. Although the idea seemed quite simple in the beginning, but making it into a reality has required sophisticated science. The earliest demonstration of an edible vaccine was the expression of a surface antigen from bacterium Streptococcus mutans in tobacco. The examples are the first successful human trial for an edible vaccine was conducted in year 1997, in which volunteers were fed transgenic potatoes which possessed the B subunit of the E. coli heat labile toxin responsible for diarrhea. Plants used for edible vaccines are like tobacco, potato, banana, tomato, rice, carrot, corn, muskmelon, soybean, etc. Preparation of edible vaccines. Live attenuated vaccines are prepared by grown disease causing organisms under special laboratory conditions that causes loss of virulence or disease causing properties. The attenuation is obtained by heat or by passage of the virus in foreign host like tissue culture, cells or embryonated eggs. Cell culture is required for the viral vaccines because viruses are replicated inside the living cells. Inactivated vaccines are produced by killing the disease causing microorganisms with heat or by chemicals. Vaccines are also produced by gene techniques where single gene is expressed in foreign host by cloning. Mode of action. The goal of oral vaccination is to stimulate the mucosal and systemic immunity against pathogen. Edible vaccine when taken orally undergoes the mastication process and majority of plant cell degradation occur in the intestine. The breakdown of edible vaccine near PP consisting of 30 to 40 lymphoid nodules on the outer surface of intestine and contain follicles. These follicles act as the site from which antigen penetrates the intestinal epithelium, thereby accumulating antigen within organized lymphoid structure. The antigen then comes in contact with M cell. M cell passes the antigen to macrophages and B cell. These B cells activate the T cell to provide immune response. In this way, immunity is activated by the edible vaccine. Here, advantages and disadvantages of edible vaccines. Advantages and disadvantages. The plants producing edible vaccines could be grown in third world countries. Plants are living organisms that changes, so the continuity of the vaccine production might not be guaranteed. The advantage is plants are regularly used in pharmaceuticals and there exist established purification protocols, and the disadvantage is the edible vaccines could be mistaken for regular fruits and consumed in a large amount then might be safe. Growing plants is much cheaper than producing vaccines. Disadvantage is the dosage of the vaccine might be variable. For example, different size bananas will contain different amounts of vaccine. Plants can host most human pathogens, so the vaccine will not pose a danger to humans. If the vaccines were grown in fields or are on trees, security would become a big issue. Importance of edible vaccines. It is used for cancer therapies like colon cancer and cervical cancer. It is used for autoimmune disease like type 1 diabetes and multiple sclerosis. It is applied for many infectious diseases like AIDS, tetanus, smallpox, measles, plague, foot and mouth disease, tuberculosis, influenza, etc. These are the reference links that are used. I am thankful to our President, Honorable Dr. Shivajirao Jondhale Sir, Secretary and Honorable Dr. Geeta Khare Ma'am, the Principal Dr. Nitin Mohire Sir for encouragement and guidance for efficient use of social media to spread knowledge through the making informative videos. Please like and subscribe our channel for sub informative videos. Thank you.